Thank you for visiting my poster. My name is Uxía Veleiro and I'm going to present you our work in GeNNius. So, as you can see, GeNNius is an ultra-fast drug-target interaction inference method based on graph neural networks. GeNNius falls within the field of drug repurposing, where one wants to find new uses for already proven drugs. As you can see in the title, GeNNius uses graph neural networks with the task of predicting links between drugs and protein nodes. As you may know, GNNs use the network topology and features. In this case, we use features corresponding to drug and protein nodes. As you can see, for drug features, we use 12 well-known molecular descriptors, and for protein nodes, we include a vector with a ratio of 20 different amino acids. In this work, we use 8 different drug-target interaction datasets, which are well known in the field. Now, let's zoom into the method section. Here you have a brief description of GeNNius architecture. GeNNius inputs the graph topology and the node features. After 4 stage layers, it outputs the embedding of the nodes. After that, we perform a concatenation of node embeddings, both a pair of drug and proteins, and pass each of these concatenations through a 2 neural network classifier. This is the edge classifier. The output will be a 0 or a 1 depending on whether there is a connection or not. This is whether this drug interacts with this protein or not. Here in this section, you can find a brief description of the model configuration as well. All these hyperparameters were selected using a grid search. Further, you will find a schematic of the message passing scheme and how it works on heterogeneous graphs in PyTorch geometric. Finally, we have a setup explanation for the experiments. With this, I can explain you the results. First, we evaluated the model using the area under the receiving operating curve using 8 different datasets. As we see, our method is achieving high AUC values while being ultra-fast. It takes less than a minute to perform training, test, and validation in all datasets. However, here we are missing a comparison because we need to compare our model with the previous state-of-the-art methods. So, in order to do that, we present the following plot. Here in this plot, we use the datasets DrugBank and VSNAP because they are the largest ones. First, we compare the model with 2 of the self-machine learning models, such as logistic regression, LR, and random forest, RF, using the same features for drug and proteins. With this, we can check the increase in accuracy due to GeNNius architecture compared with the features alone. These act as naive baselines. Further, we compare our method with 4 state-of-the-art machine learning models. We choose different technologies. These are DTI-net, based on matrix competition, Moltrans and hypertension DTI, based on transformers, and EEG DTI, based on graph convolutional neural networks. Our model outperforms them considerably, especially when it comes to training and testing times. If we look at the figure on the right, it shows how GeNNius outperformed the other models in execution time by 3 orders of magnitude. Take into account that this plot is in logarithmic scale. Further, we wanted to assess the prediction capabilities of our model, as I show you in the next plot. Datasets were released in different years and even curated in different manners. Then, for each dataset, we can find those non-interacting pairs that are annotated as interacting pairs in the rest of the datasets. Then we use those edges to validate the model, ensuring that they do not appear as negative when training the model. We obtained remarkable results showing that GeNNius could predict most of the 80% of DTIs in most datasets. The David's dataset was the worst performing one, which may be because it's highly different from the others in topology. Now, to finish with our explanation, we have 2 remaining experiments. First, our model has a given purpose, to find drug-target interactions that can be later validated in an experimental wet lab. Then we need to ensure that our model can generalize. We assess GeNNius generalization capabilities by training in one dataset, here as columns, and testing in the rest, as rows. We see how our heat map shows darker blue colors, meaning that in general our method generalizes well, especially when trained in large datasets. And now, as the final result, we wanted to check a little bit on the interpretability of the model. To do this, we generated a t-SNE, a dimensionality reduction technique, for edges both in the feature and in the embedding space. We found that the GNN was able to maintain biological information such as protein family after the convolutions. Further, we check how it spreads this biological information through nodes. In the left plot, we have a t-SNE of the protein features showing almost no group of protein nodes, here coloring by enzyme classification. However, after we use the graph neural network and obtain the embeddings, the plot shows how we now find the protein family group clearer. As an example, this kinases in pink. Thank you for watching. See you in the ISMB conference.